Enzymes definition in nomenclatural classification factor application to enzyme activity side of coenzyme. Enzyme inhibition unit of enzyme, enzyme and partner. This is a topic. Enzyme help in the specific function of our vital of the operation of over all the healthy human body. Essential for respiration, digestion, muscles, nerve function, and more. Containing 1000 mg. Human body is 1000 mg produces a specific chemical reaction within the cell. The cell can reduce some coenzymes as acceptable role of the play in every part of the body. The basic majority enzymes touch of the source or protein through the scheme of ribonucleic acid RNA molecule. RNA molecule information for DNA created protein. Enzyme which supplies specific routes of body. The help chemical reaction that the person of alive the well. The function of metabolism, the process breaking down food in drink into the energy. The enzyme catalyzes or speed up chemical reaction in cell more specific and lower than those of the touch of the source necessary to the start of the intended reaction. Nomenclature classification. Nomenclature and classification. Enzymes are named by adding the subclasses as subtract or act over lactose and lactose. Classification International Union of Biochemistry IUB classifies enzymes into 6 major classes based on the reason the catalyst. Acetoreductase, transferase, hydrolysis, leachase, isomerism and leakage. Factor affecting enzyme activity. Temperature, pH and concentration activity. First, temperature pH enzyme in optimum range estimates change in linear the protein structure. Concentration was a chapter enzyme concentration after reaction velocity. Active shade is a specific region of enzyme. A molecule blend undergoing chemical reaction. Coenzyme and isoenzyme. Coenzyme small non-protein organic molecule. Isoenzyme multiple from same catalyzing same reaction but differ the physical chemical property. Chemical enzyme inhibition substantial decrease enzyme activity, competitive non-competitive or uncompetitive. Diagnosis partner measurement enzyme level blood to help diagnose specific disease. Level to transmitted ALT, AST. Damages or amylase pancreatitis. Practical application. The modular control with fundamental laboratory tests, genetic tests, and heat covalent tests. Enzyme provides support mainly important process within the body. The digestive system and the DNA replication. The digestive system breaks down the complex molecule, small molecule, glucose to the body is useful. DNA is divided in the copy of DNA sequence genetic code. And enzyme play an important role of the processor and maintaining the Indian coil liver system. The liver breaks down toxic into the body to the range of coenzyme to attach in other activity enzyme cell. Hormone production, cell regulation, muscle contraction, material transport and respiration, signal transportation. The type of digestion enzyme and how they work. The different digestive enzymes is said. They include amylase salt. The enzyme break down carbohydrate into the simple sugar molecule. Amylase break down glycosidic bond and carbohydrate molecule. Lipid transformers. The enzyme break down triglyceride very free fatty acids and glycerol is for vital of the metabolism. lipases down the ester bond of a trickle site. Prototype process. Two type of proteases. Essential for cellular process include the cell growth, hormone signal, and formation of bioactive peptide. Perfect condition. Emission can only greater condition. For example, emission optimal temperature range in the lower temperature make it kill to work, but more than slowly. Environmental acidic alkaline. Emission changes shape in altered shape side of the substructure cannot to bend. Resultant process called denatured. Different emission and different level of activity. Mainly value environmental pH 6 to 8 touch source. The worker best in the more alkaline pH greater than 8 or more than acidic pH lower than 6 environment. Cofactor system cannot do unless the attached specific non-protein molecule now the cofactors. Two type of touch source. Cofactor ions is coenzyme. A enzyme ensures the function. Organic molecules that are also lastly bond of allow enzyme to it is a job. So cofactor thickly with a coenzyme another process group. Enzyme inhibition to ensure the body enzyme function properly. Sometimes the necessary slowly down enzyme function for instance. The body needs to stop of production several factors can limit enzyme activity level are include competitive and non-competitive inhibition and uncompetitive inhibitor and irreplaceable inhibition. Competitive to inhibitor molecule in black so that subtract compare with the inhibitor to the attached to the coenzyme. Non-competitive inhibitor molecules enzyme some activity and reduce how it is work. In computation, including enzymes and the substrates leave the activity side less easily which slow the reaction. Irascibly inhibitor. This irascibly inhibitor which enzyme then fundamentally inactivated. Explain the specific enzyme. Thousand enzymes human body perform a thousand different functions. Follow the table of Samnasam. Enzyme are coenzyme group function. Enzyme are lipase amylase. Lipase function help in the digestion record. Amylase help in danger charge into the sugars. Maltose break the sugar maltose into glucose. Trypsin break broken down into amino acid in the small intestine. Lactase break lactose the sugar in milk glucose galactose. Acetylcholine interest breakdown in neurotransmitter acetylcholine into nerve into muscles. Helicases. Unreliable DNA polymer synthesis DNA form diacrylribose. Emission pattern active liver disease. Question is emission pattern. Table come to help ALT, alcohol toxic hepatic biliary tract obstruction, liver cirrhosis, maltose, liver COVID infection. 19 valve non-such charge illness increase the MCM of the liver, ALT, AST, DLT, and CK. Creatinine CRB14 in the neutrophil count, decrease the lymphocyte count in the thrombocyte count. In contrast, no significant increase like liver enzyme for the neutrophil count survival in this group COVID. The contractive signification increases enzyme, liver enzyme. Type of enzyme: oxidation, transferation, hydrolysis, ligase, isomerase. Mechanism action: enzyme substrates called colloid reactor intermediate called MCM subtract complex ES. The ES complex produces substrate MCM and ES with pH formation ES complex afford lower activity energy. A chemical reaction B take place because creating fraction of the subtract energy to activate uh, condition called transmission state. Transmission state separating reacting produce in a chemical reaction properly concentration of reaction state. Thicker substrate transition state products the reaction is sometimes bind in the substrate, there will be no further reaction. Emulsion not only recognize the substrate but also include a formation transmission state. The emulsion stabilizes transition state. Energy change. For ES EB HTU higher into direction reaction. The transition state and difference. Activity state. A subtract bind in the side in the MCM. The formation ES complex occurs at the specific location and surface of MCM molecule. The binding the transformation substructure production occurs. The method implementation side of radiation side chain R group begin the part of the Weber MCM. Amino acid according to side of the hydrolysis group sulfurlyl tricycle group immediate touchful group of cysteine carboxyl group of aspartic acid. Future log said Professor by EML activated processing unequal conformation and complementary of the structure of the substrate. Enable two molecules fit together in match same way the key of fit into the lock. Unfortunately for the yellow model is implemented to the catalyst side. Coastline to include that area. The coastline advantage to account the specific MCM, essential for functional group active state of the MCM, not the optimal position or promoting catalyst. Molecule bound of MCM, catalyst group of achievement favorably geometrical position to the transition, the natural MCM catalyst. MCM catalyst in the surface stabilize transition state, transformation transition and state of produce. Another one feature, active state of tips of purpose packet. The magic packet stabilizer constitution co-MCM, explicit water, reactive group, co-MCM help. MCM activity state of tipper and then AP bind. Antigen binding inside of the and bind to the AG complement for the reaction occurs. Inside activity MCM also recognize substrate but actively complementary fit and transition of state of stabilizer. Activity side of activity influence fertile. Prevent the influence in water sudden formation of stable ionic bond. Concurrent mechanism of catalysis carbo peptide active state of packet substrate of peptide can check for a C terminal for factor of affecting coenzyme activation enzyme activation. The factor active enzyme reaction subtract concentration, enzyme concentration, temperature, pH inhibition. Further factors such as shared enzyme oxidation and time activator. Affected substrate concentration, keeping the factor the pH temperature MCM concentration optimum level. Substrate initial velocity reaction regarding regular herbivoria. Variably put in the substrate concentration first order kinetic. The concentration increase, the rate of increase, slow down mixed kinetic. The zero-order reaction with velocity independent of the substrate concentration. First, we must MC molecule is settled with substrate molecule to the further increase the cannot result increase the reaction. Michael-Menten derivative equation explain the Michael-Menten concentration Km subtract concentration curve maximal velocity and explains mode of parallel formula. Linear Burger plot. Michael-Menten equation can be algebraic transformation in more useful way the plot with experiment data. Receptor path S and V of the Michael-Menten equation. The plot of the 1 bar V versus 1 bar V SS, 1 bar double receptor yield the stress line. The line interpreter acts at the 1 by Km and Y acts at V 1 by Vm. Significance of Km. Km value of very without subtract. Emission will be Km very low, will high degree affected allowing more than accurate determinant of the V mass and Km. The picture first of MCM concentration increases MCM concentration will be always increase the reaction rate. Efforts of temperature. Temperature T rate of reaction optimal temperature. Activated temperature increase by greater bound marks. MCM inactivated at the temperature about 60 degree. Optimum temperature, maximum temp. Now the which reaction rate of maximum now of optimum temperature. Efforts of pH. Most MCM character pH which they are active to is maximum. Below the pH activity in glycerin. The rate of trypsin pepsin reaction velocity of pH. A subtract reaction activity mixed to the yeast is not a format. Very high pH will be HH plus will NH and loss of positive charge. The non-competitive inhibitor normal rate. The MCM is does not the yeast complex. Yeast complex does not decompose at normal rate of yield produced. Efforts are not reversibly increase the substrate concentration. V mass is lowered, Km is not alerted. Uncompetitive inhibitor blender no free MCM, but MCM is substrate yeast complex. The feature is MCM inhibitor mechanism, MCM plot. MCM inhibitor mechanism, competitive, non-competitive, uncompetitive. Equation and description. MCM inhibitor use. MCM inhibitor internally use. Sulfatrak and anti-inflammation, pseudo substrate competitive inhibitor. Produce inhibitor accumulate disease. Black brain content to produce inhibitor. HIV produces critical of a cycle of HIV. HIV produces homodimer. Inhibitor used to human aspirate producer non-monomeric. Para-amino benzoic acid. The reaction bacteria need PA for the biosynthesis of folic acid. Common clinical enzymatic pattern. Heart disease diagnosis level creatine kinase CK, lactal dehydrogenase LTH, and cardiac turbomin. Liver disease AST and ALT indicated the hepatocyte damage. ALB increase the observation jaundice. Bone disease, ALB marker, the bone disease like a Beckett disease, decay and bone tumors. Muscle disease, serum creatine kinase CK, a leveled project muscular muscle injury. Pancreatic disease, high serum amylase serum, indicator actual pancreatic. Prostate cancer, acidic prostatocranial mesothelioma PSA is now more common. Function yes, reversibly, yes, irreversibly E plus B. The Emerson band in chapter yes complex reaction. Yes, it runs rate of the reaction until the reaction is maximum velocity. Discovered the enzyme. The enzyme then ready repeated catalyst step. Equilibrium reaction instead of a free enzyme free indicated conservation the effect of reaction rate. The energy pump is much surrounded by reaction activation energy. The highest energy reaction diagram. Unable for information to subtract reaction. Internally add energy reaction instead of lower transmission energy record less activation side. The enzyme and inhibitor in functionally. Inhibitor and bind the enzyme competitive, non-competitive and uncompetitive inhibition. Summary enzymes large in the to a function of the human body. Enzymes work in competition molecular starter chemical reaction. Greater pH level temperature. Vital role of the proper function of the digestion system, the nervous system, muscles and more.